Do is go over some of the principles in basic science that have really underpinning the biology and the biomarkers and many of the findings that you've heard throughout the last day or so in bladder and, of course, in prostate cancer as well. These things are very applicable. So these are really fundamental ideas and procedures that have been used to discover the scientific findings underlying almost everything you've heard about so far today. The way this works is you take technologies, and they're listed here, such as PCR, DNA, RNA, microarrays, Western blots, and I'm going to go and explain these. These are basically used to identify the function and the relevance of genes and proteins such as FGFR3, P53, and lead to tests such as the BTA and NMP22. And again, as you heard earlier from Peter Black, these are not the be-all and end-all tests, but I want to show you paradigmatically how these were discovered, because many of the tests that do work are based on protein and DNA technology. And these, of course, are used in diagnosis, staging, and treatment. So the first question is, so cancer is driven by uncontrolled meiosis, uncontrolled mitosis, cell rupturing and death, low Warburg effect, and none of the above. Okay, so yes, cancer is a genetic disease, and it's basically a disease that leads fundamentally to uncontrolled growth. That's really the major characteristic. Cancer is a genetic disease, and basically, if this is a normal cell really has signal transduction pathways, basically the cellular machinery inside the cell that transmits signals, as you can see from some of the outside receptors on the surface, throughout the cell via various proteins, and then onto a promoter. And then the promoter regulates gene expression, which makes an RNA protein that then gets put out of the cell or inside the cell with autoregulation internally. And this is basically how the cell autoregulates itself through positive and negative feedback. On the other hand, the cancer cell doesn't have those negative feedback loops. So as a result of mutations in various of these components, the cancer cell no longer can stop positive signals from the outside or from the inside, such as oncogene, I'll show you that in a minute, to keep going, and therefore leads to uncontrolled cell division called mitosis. So there are many things that we could use as reagents really to discover how cancer works. And there are many materials, and I think of it, the analogy with the Wright brothers plane. You have the blueprint, which is the DNA. You have the RNA and the protein, which is basically the model of the plane. And then you have the metabolome, which is basically the plane actually flying. So all these things offer complementary aspects of understanding cancer. None of them are mutually exclusive, and depending on what you want to find or what you want to use for a diagnostic or a prognostic factor or target of therapy, you need different parts of this. So obviously, those are the materials, and the sample types are also varied. You use normal tissue, tumor tissue, and body fluids such as blood and urine. So the next thing that you can sort of conceptualize is the following. So when you do those analyses on the samples that I just showed you in the previous slide, you can do a number of things. So germline and host evaluation, that basically looks at the person that has the cancer. And that could be looked at using multiple techniques, microarray based or sequencing based. And then you could look at the tumor and body fluid. And this has been, and you've seen many of these examples throughout the last few days, by various technologies, RNA expression, mutation analysis, copy number, IHC, immunohistochemistry for protein, And then immune checkpoint infiltrates. You just heard from Ashish about some of these markers, and Peter Black as well. So you take all this and then you basically come together with an integrated analysis that gives you a molecular blueprint between the tumor and the host, giving you a lot of information about this. And in discovery studies, these technologies can be used for predisposition marker development, prognostic marker, predictive marker, and the predictive markers in many cases are targeted targets for drug development. So these are some of the things I'm going to show you in the next few slides, examples of how this technology was used to develop these for bladder cancer. And of course, we hope that these are useful in clinical management. And importantly, this process is reiterative. So when you have a tumor recurrence, you repeat the process to profile the tumor to see if what markers come out. So, okay. So this is the next question. PCR is preemptive cellular reaction, polymerase chain reaction, detects cancer cell growth and reactivity to therapy, amplifies specific DNA in a cell, amplifies specific proteins in a cell. Okay, so great. Well, okay, the ARS question didn't show up, but I originally had the slide. I should have mentioned that. I apologize. You basically pick any that apply. So it was more than just one. Thank you, David. So that's terrific. There you go. Yes, that's great. Well, PCR is one of the most, it's hard to say, there are Nobel Prizes and there are Nobel Prizes, okay? So this is one of the Nobel Prizes, which is a Nobel Prize that has, enormous, immense fundamental and clinical application, because it's basically the root of almost everything we do in genomic medicine currently. So it has had beyond transformative advance. So I'm going to show you a little video of how this really works. It's incredibly clever and shows fundamentally what you can do when you actually do fundamental science with no possible clinical impact. So Kari Mullis basically made a fundamental observation from putting two very basic things together. Number one, he looked at bacteria that thrive in very hot environments that have certain RNA polymerases, and then looked at replication of another organism that replicates its DNA, and combined those two features to be able to vary the temperature, and by varying the temperature using cycles from hot to cold, amplify DNA. And he thought of it as a basic science finding with no clinical application, but obviously amplifying DNA could have major impact, and it has had incredible impact. So here you go.
A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq polymerase. The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Taq polymerase is derived from hot springs bacteria and can tolerate the intense heat of a PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 35 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds, or anneal, with their complementary sequences in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine, A, pairs with a thymine, T, and a cytosine, C, pairs with a guanine, G. In the next phase, the temperature is raised to 72 degrees Celsius. Taq polymerase functions optimally at this temperature and begins polymerization, adding nucleotides to the 3' end of each primer attached to the DNA strand. After one complete cycle, there are 2 double-stranded copies of the target DNA. The PCR reaction mixture contains many copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence. Note that only 2 of the double-stranded copies consist of just the target fragment. The others also include flanking DNA regions. As the number of cycles increases, the products consist of a greater proportion of fragments with just the target DNA. After 4 cycles, half of the fragments consist of just target DNA, and half of the fragments also contain flanking DNA. With each additional cycle, the number of copies of our target sequence doubles.
So that's basically how PCR works. So the next question, choose all that are true. So SNP, pronounced SNP, is a new vasectomy technique, superior negative probability of a diagnostic test, detects differences in DNA sequences between people, single nucleotide polymorphism, detects differences in RNA sequences between cancer cells. Okay, so wow, you guys are terrific. Most people got this. 3 and 4 is the 2 correct answers. So the SNP is really the corners, the difference of single nucleotide polymorphism is really at the core of GWAS studies, which some of you may have heard of, genome-wide association studies. So basically what these are, as is shown in this cartoon here, people are different they have basically different SNPs. And these are just a hypothetical example. And what these studies do essentially is they look at the differences between DNA sequence among individuals, between patients, say, that have bladder cancer and patients that do not. And of course, there are other differences between those 2 patient populations, smoking, etc. But those are adjusted for. And then the idea is to discover if there are germline changes. So basically, germline meaning DNA changes in the DNA that makes the person that predispose you to bladder cancer. So here is a compilation of many of the GWAS studies that were done in bladder cancer. And there's a website that does these compilations. And these are the alleles, basically the piece of DNA that is different among people. And this is the gene that is next to it or implicated in that change. And this is the risk, the increased risk, if you will, of getting bladder cancer if you have that specific DNA mutation. And just a point of note is just because you have that, that doesn't mean that there's not a lot of work to be done to figure out what the mechanism is between basically having that change and why you get bladder cancer. That's not that does not come with the GWAS study. There's a lot more work that needs to be done to figure that out. This is purely an association. And you could see that there are differences here of specific alleles and SNPs that increase your risk of bladder cancer. Unfortunately, the reality of the matter is that especially when you correct for other risk factors, these things do not allow you to identify very high-risk groups of patients based on germline changes. So nevertheless, this has been more successfully used in other cancers. So the other thing I wanted to go over with is basically how the molecular biology of bladder cancers can be used for diagnostics and therapy. And this is a cartoon that has been worked on and enhanced over the years, but the fundamentals of this are still true to this day, although some of these ideas started in the mid-90s, that essentially there are two pathways towards bladder cancer development. One that is mediated by the FGFR3 RAS PI3 kinase, and another one that is P53RB P10, leading to muscle invasive disease, whereas the previous one is essentially non-muscle invasive disease. Again, there are a lot of generalities I'm making here to make the points that you'll see in the next few slides. So the two genes that have been implicated with a non-muscle invasive disease are FGFR3 and RAS, both of which have mutations with a high frequency in bladder cancer. And you've seen from Peter Black's talk earlier that those are some of the genes that are found in the mutation based tests for detection of bladder cancer, because they're very commonly found and mutated in bladder cancer. Both are signal transduction molecules. One's a receptor and one's an intracellular molecule. FGFR3 is particularly interesting because it was not discovered as a cancer gene. It was actually found to be a cause for achondroplasia, causing the fusion of the bones prematurely. In cancer, of course, it leads to uncontrolled firing of the FGFR3 receptor when it is mutated, leading to proliferation and growth. But the interesting thing about it is that many studies have shown that the mutation is actually associated with a better outcome of patients with non-muscle invasive bladder cancer. And I'm not going to go into these studies, but I've listed them here basically to make the point that this has been generally associated with a better prognosis disease. And one of the studies that is shown here is also important from the point of view of detection, because not only is it associated with better disease, but if you're going to have a test that detects bladder cancer based on FGFR3 mutation, you want to make sure that the recurrent tumor has a high likelihood of still having a mutated FGFR3, which is what this study shows. So this is a sequencing of FGFR3, RAS, and PIK3 kinase, basically between primary and the recurrent tumor in the same patients. And you could see, even from the back of the room, that the colors are the same between these two groups, meaning that you can viably use a test that's based on mutations in these three genes to pick up a recurrent tumor. Because hypothetically, if these pie charts were very different, then you actually would not be able to use those as a detection method for recurrent disease. The other thing that came out of these studies that was very interesting was the fact that they were demonstrating for the first time the presence of these two molecular pathways, the FGFR3, the RAS, versus the P53. So you could see this by the various pie charts. This is a primary tumor that was low grade, essentially, and this is a tumor that was primary but high grade. And you could see they have very different profiles in these non-muscle invasive associated mutant genes. But the primary tumors that were muscle invasive looked a lot more like the high grade non-muscle invasive disease, suggesting, in fact, they arose from that subgroup of cells. So the point is that high-grade non-muscle invasive disease is not a good thing and is more like molecularly to a muscle invasive tumor. You just happen to have caught it early. So the next ARS is choose all that are true regarding what is called a Western blot. So the Western is a foundational technology that ELISA is based on, is a molecular technique developed in Colorado, cannot be used on animal tissues, detects DNA in a cancer cell, is used to detect specific proteins in a cell. Okay, very good, very good. So yes, foundational technology that the ELISA is based on. I'll show you that in a minute. And also it's used to detect specific proteins in a cell. Okay, so this is a Western blot here. So you do essentially a protein extraction, and you denature the proteins, and then you pipette them down this electrophoresis gel, and then the proteins separate based on migration in this gel, and they look like bands like this after the gel is stained with a specific antibody. And you quantitate the amount of protein that the antibody detects. This is very similar in concept, and matter of fact, the ELISA is based on the Western, because the ELISA is an antibody that's at the bottom of the plate. You add your sample, and if you're looking for, say, PSA, the PSA is going to bind to antibodies on the bottom, and then you come back with the same antibody but is labeled, and it will detect basically the PSA molecules bound on the bottom antibody. So essentially, it's in a Western, a double capture Western in a well. So this technology is the underlying technology that is underlying the BTA stat and BTA track. Another development that's unrelated to the Western, but moving on, is the FISH technology. So fluorescent in situ hybridization underlies another technology of detection of bladder cancer, the Eurovision. I'll show you that in a minute. What this does is basically have fluorescent DNA probes that detect chromosomal abnormalities, which are common in cancer, and detect specific genetic changes in a cancer cell. So here is sort of a cartoon describing this, the Eurovision. And so it has several fluorescent probes looking at the centromere on chromosome 3, 7, and 17, which are often abnormal in bladder cancer cells. And then it has locus-specific probes at specific spots in the chromosome that are lost in cancer or gained in cancer. So you apply these fluorescent probes, and then the slides are looked at just like a cytology by a human, and then scored. Basically seeing if you have more than 2 copies. Obviously, if you have more than 2 copies, say pink here, for example, in this nucleus, that's obviously a cancer cell because you're not supposed to have more than 2 copies in a normal cell. So that's the principle of the Eurovision that you've heard again about earlier. Now let me move on to P53, and this is one that is sort of the poster child of mutations. P53 is probably the most studied gene in cancer. If it's not the most, it's close, it's up there. And it's usually altered by point mutation. There are a lot of point mutations in P53, and many of these result in extended half-life of the protein. And that extended half-life is actually detectable by immunohistochemistry, which I'll show you. And of course, the mutation is detected by DNA sequencing. So this is an example of the data from the IMPACT study, which was done at Memorial Sloan Kettering, and they've sequenced almost 10,000 patients for a variety of genes. But this is the data on P53, and this is the Kaplan-Meier curve of survival. Again, you see the number of cancers here, and some are hidden by this other chart. There are a lot. But a lot or not, the point is that mutant P53 is not good for you. And of course, it's a very heterogeneous group, many types of cancers, many different treatments. Nevertheless, P53 is not good for you. So mutation is not good for you. So that coupled with immunohistochemistry, and now I'm going to focus more on bladder cancer. Immunohistochemistry really, if you will, is like a Western except on a tissue slice. You go in with an antibody that goes with the protein, that detects the protein, and you could see P53 normal. P53 that is not mutated generally has a very short half-life. When it's mutated, it has a longer half-life, and you could detect it. This is an example of P53 mutated, very dark nuclei compared to light brown. So this is a very old study from John Stein and Don Skinner showing post-cystectomy stratification based on P53 negative or positive by immunohistochemistry. But subsequent studies and a large trial that was based on this finding that then assigned patients to chemotherapy based on p53 status, unfortunately did not reveal any difference in outcome, which was a great disappointment, especially that this trial took about 10 years to accrue to. Nevertheless, it highlights the fact that when you look at a protein at the immunohistochemistry level for its presence or absence, that loses a lot of resolution in terms of the refinements on the mutations, because the mutations occur throughout the protein, so not every mutation is the same, has obviously the same biological impact. So finally, some of the later technologies are Affymetrix, and what this is basically a detection of genes on a very large scale, essentially looking at all the genes in the genome. And the immediate application for this is for biomarkers. And this is a Affymetrix, an examination across multiple tumor sites, tumor types, and the investigators identify a cluster of genes that are expressed in specific tumors. And this is now commercially used to identify the original histological source from tumors of unknown origin. So patients that basically present with a metastatic tumor and it's not obvious where the primary is. So wrapping it up with next generation sequencing, the next generation sequencing obviously has been a landmark advance in cancer in general, and many papers have focused on bladder cancer in the past 5 or so years. And this has led not only to biomarkers of detection and diagnostics, but also targeted therapy. And I want to show you one final example now of how a mutant protein has been used successfully, probably the poster child of targeted therapy that actually, I hate to use the word cures, but leads to very, very, very prolonged remission in a cancer. And that's basically a Gleevec. And the basis of that is, as I mentioned a minute ago, is a drug that really attacks mutant proteins that are destabilizing really the signaling cascade in a cancer. And this one is specifically in BCR-ABL, and you'll see that in the following video.
Chronic myeloid leukemia, or CML, is a malignancy that leads to massive increases in the number of white blood cells in the body. Research into the specific cause of CML has led to a new approach to fighting this type of cancer. In 1960, an abnormal chromosome was identified as a key marker for CML. This chromosome, called the Philadelphia chromosome, is the result of an exchange of genetic material between chromosomes 9 and 22. The Philadelphia chromosome contains a fused gene that encodes an abnormal protein called BCR-ABL, which later became recognized as the basic abnormality in CML. The BCR-ABL protein is activated by ATP, which is a form of energy used by cells. Once activated, BCR-ABL turns on signals within the cell that lead to an abnormal increase in cell production and a decrease in cell death. A novel agent called Gleevec, formerly STI-571, is designed to specifically inhibit the abnormal signals released by the BCR-ABL protein. It does so by blocking the binding of ATP to BCR-ABL. It is believed that in the presence of Gleevec, the BCR-ABL protein is unable to function and cells revert to their normal state.
So basically, that concludes my talk. Obviously, we live in an incredible time in cancer biology. We're really at the tipping point of, I think, changing the natural history of, many, many different cancers with immunotherapy, with targeted agents and combinations thereof. So, it's a fantastic time, and, I think many of us really feel privileged to be part of it. So thank you.